as we get into another chapter, it seems like in chapter 2, we zoomed all the way down to the atomic level. And we were most recently talking about chemical bonds and molecular bonds. And I'll just bring this over here for reference because you can see our non-polar molecules still refuse to mix with the polar molecules. And as we zoom out, we're going to go for larger molecules today. We're going to talk about 4 biomolecules. Biomolecules are going to contain carbon and hydrogen. And there are 4 of them that we're going to spend a lot of time talking about. I'll grab... These will be good colors. Let's make the first biomolecule that we talk about. Let's see how this one writes. Is that okay to see in the back of the room? Some hesitation? No? Okay. We'll go with this one. Carbohydrates is one. The second one that we're going to list is lipids. Third one we can list are proteins. And the last one will be nucleic acids. So 4 of those biomolecules. We're kind of in this level where we're about to go from larger molecules to organelles. And in Unit 2, that's when we get into our first cells. So we're almost into cells. We've listed on the board our 4 biomolecules. In case we have to click through some different slides. We will start really, before we talk about any of those individual biomolecules, we're going to talk about some basic chemical reactions, how we build those biomolecules and how we tear them down. All of them, for one of our first notes, are going to have both carbon and hydrogen. And there's something kind of magical about carbon. Using 2 different colors. We were drawing these last time when we were drawing the atomic structures of things like carbon. We said it had 6 protons and 6 electrons. So in the first shell around carbon, with 6 electrons to pass out, only 2 can fit in the first shell. So then that takes us out to our second shell, which we remember has room for 8 electrons. We've only got 4 more to pass out. So when we spread out those 4 electrons, we said carbon was kind of in the middle of the road where it's got a vacancy on 4 sides. It's looking to share electrons. That versatility of the outer shell of carbon, that it can form up to 4 different bonds, makes it extremely versatile. And we'll see how many ways you can arrange these chains of carbons. Maybe to use a Lego analogy, these things would be like the square Legos with the little 4 blocks. They're extremely versatile. You can use them in all kinds of ways. So anyway, chemical structure of carbon, it's versatile. Maybe a good review question would be, when we're looking at these various structures that carbon and hydrogen can form, would you think these molecules that we're looking at here are polar or non-polar? Going back to our polar versus non-polar, where would these molecules be hanging out in this bottle of water? Would they be up here where the vegetable oils are, the non-polar molecules, or would they be down here with the water and the food coloring, which are clearly polar molecules? I heard 2 of it. I just didn't hear non-polar. Yeah, and remember we did our electronegativity chart where we said carbons and hydrogens have the same electronegative strength. They're pulling on those electrons equally as hard. But if we were to pit like a carbon or a hydrogen against an oxygen or a nitrogen, we're going to see the oxygen and the nitrogen are much stronger than either the carbon or the hydrogen. So in molecules where there are just carbons and hydrogens, there's going to be equal sharing of electrons. So all of these would be substances that are non-polar. We're almost to the point in this class where non-polar really means something to us. So hang in there. This slide just shows us once again how versatile carbon is. We can stick these chains of carbons together to form rings. This is a common construction of carbon, but we can see linear structures of carbon or carbon chains that form angles. So a very versatile backbone of a molecule. What is interesting about these kind of carbon backbones is that right now we can see hydrogens are the only thing stuck to them. But you have in your lecture notes on the second page a set of what are called functional groups. And you've got a little bit more detail in your class notes about each of these functional groups. This slide just shows us a picture of the different functional groups. So one of the things that you might expect to see potentially on an exam or quiz questions is being able to visually look at this chemical structure and recognize, is this a phosphate? Is this a sulfate? Is this an amino group? So I've plugged making note cards before. It might be helpful to have, this molecule or this chemical structure on one side, and on the opposite side of that note card, the name, so that you can flip back and forth. Each of these functional groups is going to be responsible for its own unique properties. And we can stick these functional groups on those carbon backbones to create molecules with different properties. Here are 4 that we're about to look at. So all have carbon backbones, but different functional groups stuck to them. Again, I just wanted to reference your list of functional groups that you've got some details about them. We're going to talk about them as we get into some of these examples. In speaking of these different arrangement of functional groups onto a carbon backbone, the back side of the front page of your lecture handout shows that when you stick those functional groups onto a carbon backbone, there are a couple of different ways in which you can do so. If the green in this case represents our functional groups, we can see one arrangement puts these 2 functional groups onto the same side of a molecule versus the functional group being on opposite sides of the molecule. If functional groups are on opposite sides of the molecule, this is what's referred to as a trans isomer, and cis just means same side. So cis isomers are going to be on the same side. Let's talk about isomer in the first place. We're going to start with carbohydrates. So for the time being, let's get lipids, proteins, and nucleic acids off the board. And I'm even going to get rid of our carbon backbone reminder. And if we're thinking about carbohydrates, we're going to list, let's just start this, in terms of carbohydrates. Maybe that's enough space. We're going to have to list monomers as well as polymers of carbohydrates. And if we're talking about carbohydrate monomers, there is a specific name that we can use that means a single unit of a sugar or a single unit of a carbohydrate. That term is monosaccharide. Mono just means one, and saccharide is sugar. So we've got single unit sugars, and then polymers. If we're talking about a polymer of a carbohydrate, these are sugars, so we would describe this as a polysaccharide. We're going to look at some examples of polysaccharides and some examples of monosaccharides. And in my example of isomers, when we look at monomers of carbohydrates, we're going to see 2 examples that have the exact same chemical structure or chemical formula. I'll just write the chemical formula first. There is one of these monomers that has 6 carbons. It has 12 hydrogens and 6 oxygens. It is going to be drawn in a hexagon shape, and we're going to call this glucose. It's probably the most common. Glucose is going to have a functional group, one of those OH groups sticking off of one end of this molecule. And then we're going to see a completely different monosaccharide, another hexagon shape. that's got the same chemical formula. It's got 6 carbons, it's got 12 hydrogens, it's got 6 oxygens. So the same number of each individual atom, but the arrangement is going to be different. That functional group is going to be sticking off the other end of that molecule. And this is going to go by a completely different name, galactose. You wouldn't think there's much difference between these two molecules, but because that functional group is on the other side, kind of like this cis or trans isomers, it's going to require different enzymes to break down one particular form. So there are different enzymes for breaking down glucose and completely different enzymes for breaking down galactose. Since they're structurally different shapes, we need a different enzyme to fit that molecule. We'll see more examples of monomers in a second. Just to look at the other example of an isomer, these are isomers that are mirror images of one another. So instead of just calling them isomers, they get the additional notation of enantiomers, or mirror image molecules. Your lecture notes has a picture of this, and I believe you also have a picture of this slide that again reminds us that structural isomers have the same chemical formula, but they have different structures. We can see one that is in a linear fashion, the other one is in an angle, but they've got the same number of carbons and hydrogens. Geometric isomers like glucose and galactose have functional groups in different locations. But if those functional groups are arranged in such a way that you get mirror image molecules of one another, then these are enantiomers. Like I said, sometimes referred to as left or right-handed versions of a molecule. If you'll allow me to diverge just for a second, before we get any further, it is worthwhile to point out when we're at this slide, since it's I think a fascinating time in biology. I'm always watching what that James Webb Space Telescope is spitting out. Has anybody seen any of those new images and stuff that's coming from that? There's something, what do you got?
What was it called? It's like this new, there was a big picture of, what was it, Phantom Galaxy?
I tried to click on that, but then it took me to like some page that was just a bunch of ads. So I think there's some traps out there where some things are trying to get you to look at ads rather than show you what that is coming up with. But you know, they're finding traces of atmospheres on other planets, and at some point I wouldn't be surprised if they were to find life on another planet. I mean, chemistry and physics are the same at this end of the galaxy as they are at the other end of the galaxy. So what is more of a question to me is that if and when they find life on another planet, is it made out of the same version of these molecules? And here's what I mean by that. When we make molecules in a lab artificially, molecules are made in equal numbers of left and right-handed versions. And you actually see that there are, I've erased it. We will talk about proteins in a second, and proteins have a certain chemical structure. And when meteors fall to the Earth, there's been some meteors that have hit where they found traces of proteins, traces of amino acids. And if you look into that, what they say is that when you find those amino acids, they're in equal numbers of left and right-handed versions of those amino acids. And that indicates that they weren't made by living organisms, at least life as we know it. Because what's interesting about life as we know it, every cell, every living organism that we found so far on the planet uses just the left-handed version of these molecules. The right-handed version of the molecule is biologically inactive. Since it's a different shape, none of the enzymes that we have fit that shape, and they can't break it down. So here's an example. In factories, we make artificial sugar. I have a bag of it in the back. I just got it. It says some bag of sugar that I got from Walmart. If it's artificial sugar, then it is made in equal numbers of left and right-handed versions of that molecule. Your body can use half of it, the other half is going to pass right through you. And that's why it's sometimes described as not being as healthy for you. One of the reasons it's described as not being as healthy for you versus natural sugar. There wasn't maybe they still do it. I think it was Dr. Pepper or something was marketing on their bottles that this is made with natural sugar or cane sugar. Has anybody ever seen that on the bottle when they market that? What that is telling you, if it is sugar that has been made from a plant, then it's been made by a living organism, and it's only going to be the left-handed version of those sugars. This is going to be a version of the sugar that your body can break all of that down. So it's said to be better or healthier for you. What am I really trying to say here? What's interesting is that, like I said, both of these molecules are made in the same number to start with, but all life as we know it uses one version of this molecule. There's nothing against the laws of physics that would prevent there being lineages of life forms that use the other side of this molecule, the right-handed versions. So that's why I said I was curious when they find life on other planets, if it's going to use, we've got a 50-50 chance of that life form using the same left-handed version of these molecules or right-handed versions. Anyway, physics, supersymmetry, these things are, it's interesting to think that it's kind of the luck of the draw. And that's also maybe one of the strongest pieces of evidence for a common living ancestor for all life as we know it. That all life uses, like we said, just one version of this molecule. To maybe use a different analogy, I would bet that if we were to get up from this classroom and all walk to Lowe's or walk to Home Depot and walk down an aisle and randomly grab a screw from that aisle, I bet that screw will be threaded in such a way that if we were to twist it to the right, it would go into the wall. And if we twist it back to the left, or counterclockwise, it's going to come back out of the wall. Would everybody probably, that's how screws as we know work. But those are just the screws that have survived to live in the year 2000. If you look hard, you can find screws that are threaded the opposite way. And if we go all the way back to some of the very first screws that were ever produced, they were produced, you didn't know which type of screw would become most common. And there were equal numbers of left and right-handed versions of these screws. But at some point, I don't know what happened, who lobbied who, or what event took place, but one of those versions of screws won out and is now the version that's sold in all of the hardware stores. So life was maybe on this planet likely one of the same things, where as life was starting, it had access to either one of these versions of molecules because both are around, but only one of those versions of the molecules won out. Anyway, interesting. I'll get past that, and we'll talk about how do we stick these monomers together to build a polymer. A polymer is going to be 3 or more of these monosaccharides, or 3 or more of any monomer stuck together to make a polymer. We'll see monomers, like we said, of proteins and lipids and nucleic acids. And we'll look at examples of monomers and polymers of each. We happen to have carbohydrates up, so we'll see that when we want to grow a chain of carbohydrates or grow a molecule, we're going to have to do what's called a dehydration reaction. Dehydration always occurs when you're synthesizing something, when you're building something. Notice we've got a short chain polymer and we're going to add to it. And the one end of the molecule is going to donate a hydrogen. The other end of a molecule is going to donate a hydroxide. Those snap together and form a water molecule. And the result is we've now lengthened this chain of individual monomers to make a longer polymer. Here's my example of that visually. That's why we group together this dehydration reaction with synthesis. Anytime I'm synthesizing something, so I've got 6 individual dry erase markers. And what I'm doing now is I'm synthesizing a larger polymer. I've got individual monomers, and I'm building a larger polymer. And each time I snap that together, think of a water molecule hitting the floor. I snap that together, now another water molecule hit the floor. Anytime we're synthesizing something, water's coming out. So now I've got a few water droplets on the floor. I've synthesized this larger polymer. So synthesis and dehydration go together. Now let's do the opposite. Let's take this larger polymer and let's chop it into smaller bits. We're going to chop one monomer off at a time. And now we're doing the opposite. So each time I chop one of these monomers off, I'm using the the water molecule that just fell on the floor, I would grab that water molecule and use it to do the karate chop. So this process of decomposing this larger polymer, breaking it down, is sometimes referred to as hydrolysis. You're using a water molecule to lyse or to split this chemical bond. So decomposition and hydrolysis are coupled together. So to do this chop, you're using up a water molecule to do that split. All right. Now we will look specifically at monomers of each of those four major biomolecules. And we've already got a good jump start with carbohydrates. We know the monomer of a carbohydrate is a monosaccharide. And if we were to just look at the chemical structures that I've drawn, there's carbons, there's hydrogens, but then, oh look, there's an oxygen. There's 6 of those oxygens. Those are going to be hogging, well, maybe I shouldn't give that away. Would you guess that those glucose molecules or galactose molecules, would those be polar or nonpolar? We got 2 options here. Would those sugar molecules be hanging out down here with the water, or would they be hanging out up here with the vegetable oil? We're split. We got a vegetable oil and we got a water.
They dissolve in the water.
Yeah, if you've ever made sweet tea, you got to mix that sugar in with the water. And the sugar does mix with water. So sugar, since there is an oxygen involved, that oxygen is going to be pulling on those electrons harder than either of the carbons or hydrogens. So we're going to have an unequal sharing of electrons. Each glucose molecule is going to have a tiny positive end and a negative end. So they're going to be able to form these hydrogen bonds. So all of these sugar molecules, we talked about glucose and galactose, but before I get any further, let's write fructose up here. Fructose and ribose are 2 other examples of monosaccharides, but if we count them, they've only got, it's a 5-carbon chain. So we'll draw it in this little pentagon shape. This is going to be fructose. And there is another isomer of that that's got the same structure, this pentagon shape, but it is going to be called ribose. The last one there, ribose, is going to be the main backbone, the sugar backbone of the DNA and RNA molecules. The first 3, glucose, galactose, and fructose, are commonly broken down as a fuel source by cells. So your lecture notes, I believe, has individual pictures of glucose, galactose, and fructose. In fact, I stole one of the slides from a physiology PowerPoint and I added it here just to show you some examples of polysaccharides, since we're about to switch gears to polysaccharides. These are sugars that are made of 3 or more of those monomers. And we're going to see how the body breaks down these polysaccharides. Either the examples of polysaccharides that we will list on the board. Let's go ahead and do that. First example of a polysaccharide I'll list as starch. And maybe to help out, I'm going to come over here and underline starch with a green color. That symbolizes that it is found in plants. We'll see 2 of these polysaccharides that come from plants, starch and cellulose. Cellulose also is going to come from a plant. But there are 2 other polysaccharides that we'll have to mention. I'm looking for another color. Maybe this color will work. That are going to be found in animals. We'll say animals and fungi, just to be clear, because there's 2 examples that we'll see these polysaccharides. One of them is glycogen. glycogen, and then down here I'll just write chitin. Both glycogen and chitin are going to be found in either animals or fungi. Okay, so we can kind of see we've got starch and glycogen at the top. These are going to be two of the polysaccharides that are used as an energy source. Starch, glycogen are broken down as an energy source. I'm going to list that on the board when I get over there in a second. But as these are broken down into smaller pieces, we can see this layer that tells us there are 2 unit sugars. I didn't form a column for this, but I'll just reference it from this slide. A 2 unit sugar is a disaccharide. We know saccharide is sugar, mono is 1, di is 2, and poly is 3 or more. So there are 3 common disaccharides or 2 unit sugars. There is lactose, which is made of a combination of a galactose and a glucose. There is the second disaccharide, maltose. It's made of a combination of 2 glucose molecules. And then you've got sucrose, which is made from a combination of glucose and fructose. Notice that each of those disaccharides has its own unique enzyme that breaks it down. And the enzyme always ends in an ase. So lactase is specific to breaking down lactose. This chemical shape is different than maltose or sucrose, so the enzyme that breaks down lactose isn't going to fit either of those other 2 molecules. Maltase breaks down maltose and not any of the other structures, and we can see sucrase breaking down sucrose. Up here there are even other enzymes. We can see the ase ending there, salivary amylase and pancreatic amylase. Maybe to just give you what's a short breakdown of what's going on there. Salivary amylase, this is the stuff. Imagine you're eating a Jolly Rancher and your mouth is getting all watery. That's salivary amylase that's produced by your salivary glands, and it's good for breaking down carbohydrates. It's good for breaking down glycogen, starch, or glucose. And it is about 10% as effective as pancreatic amylase. This is 90% more effective. It's much stronger at breaking down carbohydrates. So that's help, not that we have to get into the digestive system, but I'll say it, well, let's just say this very quickly. I can't have a thought in my brain without sharing it. So the pancreas, since we're talking about pancreatic amylase, the pancreas is going to be one of these glands that you really can't live without. So there is one of my dad's buddies that has pancreatic cancer. And anybody that has pancreatic cancer, it's not a good one to have, because the pancreas is responsible for breaking down food that in specific carbohydrates that you don't keep in your mouth long enough. So if, it was told to me one time long ago that you're supposed to chew your food a certain number of times before you swallow it. And that makes sense because the food spends longer in your oral cavity, it comes into contact with the salivary amylase and gets chemically broken down into smaller pieces. It turns out that any of these carbohydrates are going to have to be broken down into their individual monomers if they're going to be absorbed into the bloodstream. If I could just jump to the next slide real quick, it's also one from the physiology class that I grabbed that shows the yellow area is representing the lining of our digestive system. And these cells make up the wall of the digestive system. So if we were to eat donuts in here, the donut, the sugar of the donut would be up here, and it would have to be broken down into its individual monomers. Those are small enough to be absorbed across this plasma membrane. If that sounds familiar, I was saying something last time about why we needed to know polar versus nonpolar, because it has everything to do with how easily things can cross the plasma membrane. If you remember, we said anything that is too large or anything that has a charge to it isn't going to be able to cross the plasma membrane freely on its own. It's going to need some type of channel to do so. And if you look closely, here are a couple of channels that are specific. If I can plug proteins for just a second, we won't get to proteins today. It's going to be one of these 4 molecules. But if I can talk about proteins for a second, they are going to be the most, in terms of functions, they vary the most in terms of how many different types of functions they can pull off. They're way more varied than either carbohydrates, lipids, or nucleic acids. In the case that we're looking at now, these proteins are acting as little transport vessels, not unlike the door is acting like a transport vessel for us to get out of the classroom and into the hallway. So here are some proteins. We can see these are specific for each monomer. So this is GLUT5. I feel like I wrote GLUT on the board last time when we were talking about transporters. This just stands for glucose transporter. And 5, meaning it's the 5th one they discovered. There's over 20 different GLUT transporters. This one is specific to fructose. You can see there's another transporter that is going to be specific to glucose or galactose. So at some point, we have to break these sugars down to their individual monomers if they're going to be absorbed by cells and then make it into the bloodstream. So here I'm going to go back to this slide. How we get them broken down into their individual monomers is by taking the polymer and chopping it down into individual disaccharides. Here's where that pancreatic amylase is much more effective. And if you're like me, somebody that doesn't have time to choose their food 30 times before they swallow it, I might have 3 minutes to eat a burger. And if I'm biting into that burger and swallowing, then the bun of that burger never has a chance to get broken down by the salivary amylase. But that's okay. As long as my pancreas is working, it's going to be able to pump out this pancreatic amylase that's going to break down those carbohydrates. If, like we said, you don't have a functional pancreas, then you're not going to be able to break down carbohydrates or other things. Anyway. Not that we really have to know that. We do have to know, like we said, examples of monosaccharides. So fructose, galactose, and glucose are the common ones broken down as a fuel source. We need to be aware of disaccharides, lactose, maltose, and sucrose. Maybe be aware of what combinations produce a lactose, what combinations produce a maltose, what combinations produce a sucrose. But just to talk about the polysaccharides real quick, the first polysaccharide that I'll talk a little bit more about is starch. And it's going to be found in plants. It looks something like, I'll draw it like this in a green color, where starch looks like a coiled spring. And all along this coiled spring, we're going to see these individual glucose molecules. So instead of drawing them like a hexagon shape, I'm just going to simplify this and draw each glucose molecule as a little circle. So we can see we've got a polysaccharide. It's a chain of 3 or more of these glucose molecules. And when it's in this coiled little spiral like this, it's real easy to chop up. If we were an enzyme or these molecular scissors, we can go in here and just make one little snip. If I make this one little snip right there, I've now released one of these glucose molecules out into the bloodstream. So starch, I can just make one little snip right there, and I've now released a second glucose molecule out into the bloodstream. Starch is very easily digestible or chop-upable, if you want to think of it that way. That's why starch is used as an energy storage molecule. I'm going to say short-term energy. And to be specific, I'll say short-term energy in plants. That's what the green is about. Cellulose, if we can switch gears to cellulose, we're going to see that it has a different structure. It too is made out of chains of glucose molecules, but these chains of glucose molecules are locked in these lattices with one another. And it forms these sheets. Let's see if we can sketch one of those. Cellulose would look something like, I'm going to cheat and give myself a little more space. So cellulose is in plants, and what we can notice about cellulose are these sheets that form layers like a lasagna. And all along those sheets, we're going to see chains of these glucose molecules. And what's unique about this cellulose is that each chain of glucose molecules is going to form a hydrogen bond with the chain above and below it. So each glucose is locked in there with multiple bonds. That makes cellulose tough and indigestible. If I'm acting as these molecular scissors again, and I want to come snip off a glucose molecule, and I make a little snip right there, I haven't released any glucose molecule. Or if I come in here and make a snip, each of those glucose molecules is still held in place by multiple bonds. So cellulose isn't going to be used as an energy storage molecule. Instead, it's going to be used as a structural component in plant cell walls. So cellulose, we're going to say found in plant cell walls. Going back to the slide, let's switch gears and look at these polymers of carbohydrates that are found in animals. The first example that we can see up here is called glycogen. And if you look at glycogen, it kind of looks like starch. It's got these spirals, these coiled chains of glucose molecules, but they kind of form these branch points where these coils branch off from one another. So let me simplify glycogen. Glycogen, without drawing it in a coiled form, I'll just draw it in a branching form. We'll see glycogen forms these branches. And along each branch, like we were seeing with starch, there are just these chains of glucose molecules. And look what happens when it's in this branching type form. It becomes very easily digestible or easy to chop up, like we said starch was. If I was an enzyme and I came in here and just made one little snip, I've now released this glucose molecule into the bloodstream. Or we can make a snip down here and release this whole chain of glucose molecules into the bloodstream. So glycogen is easily digestible. And when we list the function of glycogen, we're going to say short-term energy storage in animals. Animals are going to store their excess glucose molecules as glycogen. It's stored in the liver. We mentioned before the liver is one of these organs that stores up extra nutrients when there's plenty. So you eat a whole tub of ice cream, that extra glucose is going to be stored as glycogen in the liver. We're then going to see chitin real quick. Chitin is another polysaccharide that is found in animals. Maybe without me drawing chitin, you can see a molecular picture of one of the chains of a monomer of chitin. It's one of these glucose backbones, but one of the functional groups we can see contains nitrogen. And that means that chitin is a polysaccharide that contains nitrogen. The two places that we find it most commonly, it's going to be found in the cell walls of fungi. So all fungus, we will learn, they too have cell walls, but their cell walls are not made out of the same stuff that plant cell walls are made out of. Plant cell walls are made out of cellulose. Fungi cell walls are made out of chitin. The other thing that chitin is made out of is we're going to see insect exoskeletons. So things like spiders or grasshoppers, any insect that has a hard exoskeleton, that exoskeleton is made out of chitin. Maybe a real life example of this is we live in an area where there's lots of bats, and if you've gone under an underpass and bats live there, you can smell that because their bat droppings have a lot of nitrogen in it. The reason bat droppings have a lot of nitrogen in it is because they eat almost exclusively insects, mosquitoes, things that they catch in the air. And if your diet consists mainly of exoskeletons of insects, then your waste is going to have a high concentration of nitrogen. It's also sometimes sought after as a good fertilizer for plants simply because it has high levels of nitrogen. And now you know where that nitrogen comes from. It comes from the exoskeletons of the insects that they eat. Okay, let's look at this picture real quick because it reminds us of where we are going. We're going to end up zooming out to talk about how things get in and out of cells. And so far, what we have seen is carbohydrates. And we can see carbohydrates sticking off the surface of a cell. Some of these carbohydrates are attached to a protein, so we call those glycoproteins. And sometimes these chains of carbohydrates are attached to a lipid, and we would call those glycolipids. So one common example of a glycoprotein would be the types of proteins that are found on the surface of your cells. Here I am plugging proteins again, and we can see proteins vary. They're all over the place. One of the things that proteins do is they act as these identifying molecules. And how they identify one another with these little flags basically that are made out of short chains of carbohydrates. And if they're flying the same flag, it's how, if you're flying the same flag that I am, it's how these two cells are able to tell, all right, I'm a liver cell and you're a liver cell. So if they identify as liver cells, then we're going to see other proteins are going to be these adhesion proteins. So they're going to be held together. It's how the body forms specific tissues. Tissues have to recognize that they're part of the same group of tissues, and then adhesion proteins will lock them together. That's the problem with cancer. We're going to see here we are jumping to Unit 3. But in cancer, we're going to see that the DNA instructions get all chopped up. So the proteins get all misformed. And what used to look like a flag that you could recognize as a liver cell now doesn't look like anything that you recognize. So if the identification proteins don't work, then the adhesion proteins are not going to work. And then you get these cells that roam around the body and don't attach to anything, and they can disrupt organ communication. And that's when it becomes bad. So anyway, cells have to be able to identify with one another. And the example, if I can jump to the lecture notes, I said we would go back and forth. We compared isomers, these mirror image molecules, which are enantiomers, and eventually we're getting to carbohydrates. We reviewed, just by looking at this, if we didn't have any labels on this at all, you were seeing that we're extracting a water molecule to build something larger. This is a synthesis reaction, so dehydration and synthesis go hand in hand. Just like decomposition, if we're going to take something larger, this is a disaccharide, and we're chopping it into individual monosaccharides, we had to invest a water molecule to do that, so hydrolysis. Carbohydrates. There's the monomers. Here are disaccharides, and we've just talked about the polysaccharides. And I was about to show this cell identification that you have. Maybe the best example would be on all of our red blood cells. These have surface identifying structures, and that's how we know, since there are blood transfusions done, we have to know what type of blood is that. Are these blood cells going to be able to recognize one another? And this has to do with surface markings, these short chain carbohydrates. So everybody has the instructions to build these 3-chain carbohydrate structures on the surface of their red blood cells. But some people have additional structures to put an additional unit on the end of that short carbohydrate chain. If there is a glucose put on the end of that short chain, it ends up being one type of red blood cell. We notate that with a letter A. If there is in your inheritance instructions, if you inherited a mutation that says, we're going to put an additional carbohydrate out there, but it's not going to be glucose, it's going to be galactose instead. That could happen, in which case you're going to end up with a type B red blood cell. Or you might have inherited a version of a gene that doesn't have instruction to put that additional monosaccharide out there at all, and you would have type O blood. we will get into more examples of inheritance and how these different versions of instructions play together. These are different versions of a gene, so we call those alleles. And sometimes alleles will completely dominate the presence of another allele. And other times they have more of a codominance, which is the case here. Again, somehow we jumped into Unit 3, but I just wanted to use an example, since we're about to get into lipids next, of how carbohydrates are helpful when we look at the surface of cells. They're going to be helpful by forming these identification structures. All right, let me see if I can get through lipids, and that might be a good time to draw the line in the sand for our biomolecules. Is it okay if I erase our carbohydrate monomers and polymers? Let's, instead of carbohydrates, let's get this out of the way and write in lipids. In fact, in lipids, we're not going to need to know individual monomers. We're going to go straight to polymers for lipids. So let me draw this another way. Clicker. We're already on the first lipid, so let's switch gears. If we're talking about biomolecules, let's now focus on lipids. Maybe we can do a little concept map here with the lipids. There are going to be 3 different lipids that we need to be aware of. The first one we're going to call a phospholipid. Maybe we can just come straight down here with the next one. This is going to be cholesterol. And the third and final one that we'll list is a triglyceride. We'll start with the phospholipid first. We need to be able to recognize structurally that we're looking at a phospholipid, that it's different than a triglyceride or a cholesterol. So let's just do that. A phospholipid we can always recognize by looking something like this, by having what I will draw as a circle and these 2 squiggly lines. What this represents, the circle represents the head region of this phospholipid, and then down here we can see the tail region. There are 2 of those fatty acid tails. We're going to draw those more closely and see why they get the name fatty acid. We're reminded of what acids do if you're thinking of last lecture. But for now, let's list that the tails are all going to be nonpolar, and the heads are all going to be polar. If I can stick with this real quick before drawing the structures of the other 2, since we're seeing how the head is polar and the tails are nonpolar, if I go back to this thing right here and shake it up just a little bit, what you see temporarily, can you see those spheres that are forming, those little balls? If we let that sit, these are all going to dissipate, and it's going to separate once again. But what happens is there's a little bit of water trapped in the center of those lipids. And lipids, since they have polar ends and nonpolar ends, if I were to fill this with phospholipids and shake it like that, we would see each of those phospholipids spontaneously form this double layer that is our plasma membrane. We said this last time. Instead of there being a moat filled with alligators around the perimeter of our cells, instead we have this moat that is basically a hydrophobic wall. These tails, remember, are held together by hydrophobic interactions. Hydrophobic interactions. So we know if you are a nonpolar structure, like these lipids, you can form hydrophobic interactions. And that's one of the reasons why nonpolar substances can just move right through this nonpolar wall. Nonpolar things get along with other nonpolar things. But if you're polar, you're going to have a tough time just crossing this plasma membrane on your own. We'll list this as plasma membrane. And I'll just say with words, since I've already drawn it here with a picture, the main function of a phospholipid, it's the main structural component in a cell's plasma membrane. Main component of the plasma membrane. And that's why we see, in addition to just that double layer of phospholipids, if we're going to get certain things through like big glucose molecules, we need these proteins to form these transportation routes for large things to cross that plasma membrane. So the phospholipid, its main function is to be the premier structural component of plasma membranes. Now that we've said that, let's jump over to cholesterol. Cholesterol does not look like this. Cholesterol instead is easily recognizable because it is made of a series of 4 fused carbon rings. We're going to see 2 of those carbon rings look like 2 hexagon shapes connected. Then there's a third hexagon. I've not given myself quite enough room to draw this. But you've got 4 of these fused carbon rings, and then there's a functional group that sticks off the end. That functional group may vary. What we need to know about cholesterol, it is the precursor molecule to all of the different types of steroids. How can I edit that down? It's, we'll say, used to make steroids. So your body takes the cholesterol and manipulates that functional group. By moving it around, it can convert this cholesterol into testosterone or into estrogen or progesterone. Here's the picture of our cholesterol. It's got a functional group sticking off of it. And like I said, we can modify the location of those functional groups to change this cholesterol into one of the different steroids, estrogen, progesterone, or testosterone. Cholesterol is a nonpolar molecule. So when we go back to this picture of a cell, look where you find cholesterol. We find it hanging out in that nonpolar moat. It would be hanging out up here with the lipid molecules. It's a lipid. It would be hanging out with the other nonpolar molecules, I should say. So when we think about cholesterol, it's worth saying that too little cholesterol is bad. So when you hear people saying, I got to take cholesterol completely out of my diet, well, you need that cholesterol to manufacture testosterone and estrogen and progesterone. You have to have some of it. But too much cholesterol, if you had an abundance of cholesterol, we can see where it would be stored. It would be stored in the plasma membrane, in that little hydrophobic moat. And an abundance of cholesterol makes this plasma membrane too rigid, and it makes it difficult for things to freely cross that plasma membrane. So too much cholesterol is bad, makes it difficult for things to cross the plasma membrane, but too little cholesterol is bad. You wouldn't be able to manufacture the needed levels of certain steroids. Okay, so phosphates, steroids, that leaves the last one to get into, and that would be these triglycerides. Triglycerides are easily recognizable because they have this glycerol backbone. The glycerol backbone is just 3 carbons, and attached to this chain of 3 carbons are going to be what we call fatty acid tails. Fatty acid tails. You know, instead of me, I'll just do the quick sketch that looks like this of 3 fatty acid tails. Because what we have on the screen, we have a better diagram of those fatty acid tails. And you can see where they get the name acid from. We said last time acids are anything that have an abundance of hydrogen ions and would tend to donate a hydrogen ion if put into a solution. Sure enough, if we are going to digest lipids as our fuel source, if we come in here with molecular scissors and start chopping up these fatty acids, you can see they're going to be releasing hydrogen ions into the solution around us. So anything that is likely to donate a hydrogen ion would be an acid. These fatty acids certainly will do that, like I said, if we start to chop them up. So let's compare. Let's first maybe list the function of these triglycerides. These are quite large molecules. If we can go back to the chemical formula that I'd put up for glucose earlier, we said it had 6 carbons, 12 hydrogens, and 6 oxygens. It's not a very big molecule compared to one of those large triglycerides, so it's going to be very quickly broken down. But something like a lipid, we can see just the fatty acid tails have up to 30 carbons on them, and we've got 3 fatty acid tails here. So these are really large molecules. They take a long time to break down, which is why if you've eaten a meal that has a lot of triglycerides in it, a lot of vegetable oils in it, it will make you feel full for longer periods of time, as opposed to just going and eating like some pasta that just has chains of glucose. Your body's going to break that down and you'll be hungry much more quickly than if you were to ingest some of these triglycerides. I'm not saying eat a fatty diet, but I'm just saying it will make you feel full longer because these take longer amounts of time to break down. They're just larger molecules. And if we're breaking down triglycerides, since I'm giving health tips here, which I'm probably not the authority on that, it's worth pointing out that these fatty acid tails are compared to one another as being saturated versus unsaturated. You should have this picture in your lecture notes, and you'll notice that saturated fatty acid tails, what that means to saturate a fatty acid tail, means that it is completely stuffed full of hydrogen ions. If I pulled a hydrogen ion out of my pocket and tried to stick it onto this fatty acid tail, there would be nowhere else to stick it. It's completely full of hydrogen ions. What that also does, saturated fatty acid tails, as opposed to these unsaturated fatty acid tails down here where there is at least one double carbon bond. So that means if there is a double carbon bond, so carbon bonds are sharing 2 electrons in this case, then there's room right there to put extra hydrogen ions. If I had them, I could stick extra hydrogen ions to this fatty acid and then it would be saturated. I'm about to compare saturated to unsaturated, and you can see unsaturated fatty acid tails are liquid at room temperature versus the ones that are saturated, these are solid at room temperature. The reason for that is that these saturated fatty acid tails, to use my fingers maybe as an analogy, my fingers are bent currently, and that is to represent the bent nature of these unsaturated fatty acid tails. When my fingers are bent, my hands are taking up less space. So these molecules could slip past one another at room temperature. But if we were to saturate each of these fatty acid tails, then it causes those fatty acid tails to elongate slightly. So we can see when they're completely stretched out, these are larger molecules and they tend to get stuck together. And so you'll see that saturated fatty acid tails are often locked together and solid at room temperature. So butter is a good example of that. Or if you've cooked with, Crisco that comes in that jar that you got to scoop out, that is the saturated version of Crisco. It is less healthy because when you digest that, there's more hydrogen ions that are going to get released. It's crispier, and somebody might argue better tasting, but less healthy because it's going to kick off more of those hydrogen ions. I feel like I'm saying that a bunch without doing a short little reminder that we were looking last time at the pH scale. And we said it was a measurement of we'll say it measures free hydrogen ions in a solution. And we said it went from a scale of 0 on the low end to 14 on the high end, and right in the middle was 7, a neutral pH. But our blood pH had to stay within a very narrow range. We said this was 7.35 to 7.45. And that means that if there are too few of those hydrogen ions, what really happens there, if we get too far below or above this range of free hydrogen ions in the bloodstream, all the proteins are going to change shape and denature. Has anybody eaten or is a fan of ceviche or anything like that? Anybody? Me, I like my food fried and crispy, obviously. So I've not eaten a lot of that ceviche. But what that is, it's typically meats like fish or even ground beef sometimes, that instead of cooking it with heat, they cook it with acid. They use lemon juice most of the time. And if you put proteins in a very acidic solution, it's going to cause those proteins to denature or change shape. That's the same thing that happens when you heat a protein. It denatures and it changes shape. So that's why cooking with acid is sometimes used rather than cooking with heat. But the proteins that are in our bloodstream will also change shape if the pH gets too acidic or too alkaline. So back to this picture here, one of the reasons we want to be careful with ingesting too many of those saturated fatty acid tails is it can more readily shift the pH of the bloodstream. Okay, triglycerides. Here's the last thing I'll say, and I think I have just enough time to do this, and then we'll take our short little break. We did start at 12:30. I keep thinking sometimes we start at 12, but we started at 12:30. So I got just enough time to get through this. I'm going to break this down in just 2 slides. What we're really looking at here is the digestion of one of those large triglycerides. I think of this sometimes as a table with 3 legs. Okay, each of those legs is a fatty acid tail. And when these triglycerides are ingested, they have to be broken down into a small enough piece that they can be pulled across the cell membrane. Remember, it's all about how do we get these things across the plasma membrane. Cholesterol doesn't have a problem. It's nonpolar. It can move right through that plasma membrane. Triglycerides are large. So even though they are nonpolar, they're just too big to move through this plasma membrane on their own. So I don't know if this is too fuzzy to see what's going on, but if we can just zoom into the top half of this picture, this triglyceride, as it's taken into the digestive system, is first going to get broken down into a monoglyceride with 1 fatty acid tail, and then just 2 of those fatty acid tails are removed. It's not stored like this. This is a temporary structure. These are not going to stay this way. They're only temporarily broken down this way to cross the plasma membrane. Let's go on the other side of the plasma membrane to see what happens to these molecules as soon as we get to the other side of this plasma membrane. That monoglyceride and those 2 fatty acid tails are put back together. It's stored as a triglyceride in tissues like adipose tissue. That reminds me, we should come over here and list triglycerides' function. It is a long-term energy storage molecule. So long-term energy, it's stored in tissues like adipose tissue, and your body can go to those triglycerides and break them down as a fuel source when needed. I only make this point about crossing the plasma membrane because we talk a lot about that this semester. And triglycerides are too large. So we said this membrane separates based on size and based on charge. This does not have a charge, but it's really large. And my quick analogy to see, I don't know why I think this is so fascinating, is it's another example of how cells don't waste energy. This is going to be stored as a triglyceride or as a table with 3 legs. So there's no need in breaking it down any further than you need to break it down to get it into the cell. Here's maybe a more relevant analogy to the level that we live at. So before I worked here at Temple College, one of the things I did for extra money was one of my buddies. He still works at Home Depot, and he was in charge of a crew of people that would come to your house and rip out your old windows if you happen to buy new windows at Home Depot, those energy efficient ones. And in a matter of hours, we could show up to a house, rip out the old windows, put in these new windows, and be off to the next job. On a good day, we can do multiple jobs. But what's supposed to happen when you go to these jobs is the people that have bought the windows, they know it's going to go in this room. So they're supposed to move the tables and chairs and everything that is precious to them away from the tables. One of these jobs we went to one morning, early morning, it was this sweet old lady's house that had a really nice table next to one of her windows. And she was by herself. She couldn't move that table. She actually wanted it to go in another room, but she could never do it by herself. And so I said, okay, I'll move the table for you. And all I did was take 2 legs off of the table. And it made the table kind of an L shape. So you have to imagine if we had this long L-shaped table, you and I could just move it out here by opening the door and kind of sticking the long end of the table out and then kind of finagling it around the corner this way. And then when I got it out into the hallway, I was able to snap the other 2 legs on, and the table was right back in its functional shape, and we were on to putting in windows. And my buddies didn't think anything about it, but the whole day I'm talking to them that this is exactly how triglycerides are crossing the plasma membrane. You know, I didn't waste any. I could have taken off all 4 legs and then taken the table out there and put all 4 legs back on. That would have worked, but I would have wasted some energy. I only needed to take off 2 legs. The cells have figured that out way before I was figuring that out with the table. So triglycerides, they're too large to cross the plasma membrane, just like a table with 4 legs might be too large to get out of this door. But if we take just 2 legs off, that's small enough to get it across the plasma membrane, then we can reassemble it inside the cell. Let's take a break. I appreciate you letting me go a little bit past what I intended to there. And on the other side of this, I am going to set up our lab activity. I'll wait till everybody gets back. I'll do a quick demonstration of where to find everything.